All right, everybody, we're going to go ahead and get started. This is the third and final AP Biology review, but it's not the final units. We're going to be talking, I'm going to turn on my microphone. There we go. We're going to talk about units 5 and 6. And then this weekend you'll study natural selection and ecology, which I think are the easiest of the units. So don't miss that opportunity to get amazing points on those 2 units. And then that should be it. So then we'll do a practice MCQ on Monday, and then the test is on Thursday. We do not have a practice FRQ. It's very hard to self-grade a practice FRQ, but there are FRQs on AP Classroom if you want to practice taking an FRQ, for sure. And I've been giving you practice FRQs all year long. So every test that we had, those questions were previous FRQs from previous exams. So you've had some experience with it. Okay, let's go ahead and get started then. We're talking about heredity and then gene expression. So in the heredity unit, we talked about meiosis. Last week, we talked about mitosis, that was cell division, and it was called non-reductive cell division. This time it's called reductive cell division. Can somebody remind me why it's called reductive?
Because they lose the chromosomes, there's not as
Okay, because we cut the chromosome number in half. So this is the type of cell division that's creating our gametes, our sperm, egg, or pollen, if it's in a plant. And then when they come together, they form a diploid organism. And when 2 gametes come together, we call that a zygote. Just a reminder of some vocabulary from the past. All right, diploid cells have 2 copies of each chromosome. So what's that picture, that black picture called? Anybody remember? Starts with a K.
No.
Isn't it a karyotype? Very good. That's a karyotype. So that's a photograph of somebody's chromosomes, all 46. So there's 23 pairs. And what's the sex of this individual? The biological sex of this individual. Male. Yeah, they have an X and a Y chromosome. So what we have there are pairs of homologous chromosomes. Remember, you get 1 chromosome from mom and 1 from dad for every set of chromosomes. So you have 23 pairs of chromosomes. Don't confuse homologous pairs with sister chromatids. So if you have an X chromosome, it looks like this. This side is identical to this side. Those are the sister chromatids. They're identical to each other. Then you have 2 pairs of them. 1 came from mom and 1 came from dad. They're similar to each other. So if there's a gene on this one for eye color, then on the other one, there's a gene here for eye color. But mom's might be blue eyes and dad's might be brown eyes. But they're similar, and that's a homologous pair. Okay, and then what stage of mitosis? We're going to be talking about meiosis, but I want you to think about mitosis. What stage would this be? Oh, it's right there.
Prophase.
Good job. Prophase is right. So prophase is when the chromosomes show up. Now in meiosis, prophase is different because the chromosomes are going to pair up and they're going to trade DNA. But notice that the chromosomes are all bivalent in that image. Okay, so this is meiosis. If you look all the way up at the top of the diagram, the first cell is labeled a germ cell. Those cells are the cells that divide to make gametes, so the 2 G's go together. In mitosis, the cells that are at the top are called somatic cells. Those are the body cells. So instead of instead of just IPMAT, we have IPMAT, PMAT. The first half of meiosis is denoted with the Roman numeral 1. The chromosomes cross over, so that's what's happening in prophase 1. Look at the diagram. I know some of you guys are kind of far away. Prophase 1, they're crossing over to create new genetic combinations. They're lining up side by side in metaphase 1, so metaphase middle. And then they get separated from their homologous partner during anaphase, and then telophase is when the chromosome number gets cut in half, and that's telophase 1. And then the second half of meiosis is Roman numeral 2, and it's identical to mitosis, only you're going to have half the DNA because that number got cut in half during telophase 1. So everything about it now is exactly the same. Notice there's no crossing over. They don't line up with their pairs in the second half of meiosis because their pairs are in the other cell. Their homologous partners are in the other cell. Okay, and then just a couple practice vocabulary. Homologous pairs connect to each other. That's called synapsis, forming structures that are called tetrads. And then each connection, which is called a chiasma, the DNA then gets swapped over. So that's what the crossing over is. And it's creating unique chromosomes that nobody has ever had before. What are those 4 cells at the end of meiosis again? Just a review question. Gametes is a great word. So sperm, egg, pollen, they're haploid cells. All of them are haploid. Okay, so what is genetic recombination? Any event that leads to new combinations of genetic material. And there were 3 things that we learned about in this class. Crossing over is creating new combinations. Mutations actually form new DNA. Most of the time that's a bad thing, but that's what evolution kind of counts on, is an occasional good mutation. And then 3, the random fertilization of umpteen different sperm with however many eggs. There's many, many, many possibilities for the possible combinations of chromosomes in the zygote. So that's genetic recombination. And then the College Board wants you to know the difference between Mendel's law of segregation and his law of independent assortment. Remember that the word segregate means to separate. So think of segregation in our ugly US history. So the 2 alleles get separated from each other. And then which side or which gamete they go into is random. And then his law of independent assortment says separate alleles for separate traits are passed on independently of each other as long as they aren't linked on the same chromosome. If they're on the same chromosome, he didn't know that. If they're on the same chromosome, then they do get transferred together. So to the right, when Mendel looked at 2 different traits, were they inherited together or independently? Well, that's a little bit complicated, but his results, let me see if I can get my image out of the way here. So he crossed, and remember that he can't see the letters. You can see the letters, but Mendel can't see the letters. So he crosses a yellow pea, yellow round pea with a shriveled green pea. And if they're inherited together, he would expect a 3-to-1 ratio. And if they were inherited separately, the color and the shape of the pea, if they were inherited separately, then he would expect a 9-to-3 to 3-to-1 ratio. This is what he actually got. 315 were yellow round, 108 and 101 were green or yellow shriveled, and then 32. So that is actually the 9-to-3 to 3-to-1 ratio. So as long as they're not on the same chromosome, they are being inherited separately. So that's independent assortment. Complicated. All right, Punnett squares become more and more complex as the possible outcomes increase. So better to do monohybrid crosses for each trait and then multiply the results. So this is just a dihybrid where you have 2 traits. So the trait on the left is color and the trait on the right is the shape of it. Rather than doing, give me a second. Rather than doing one of these, oh, now it just went way back. There we go. Rather than doing one of these giant Punnett squares, and it could be even bigger than that, you can just do 2 simple monohybrid squares and then multiply the results. So if you want to know what percent are going to be yellow and wrinkled, you just do 3/4 times 1/4, and you would know that it was 3/16 would be wrinkled. Okay, the test cross. Remember, it's a method of determining the genotype of an organism that has the dominant phenotype. So in this example, purple is dominant, and Mendel can't tell if this flower is homozygous dominant, has 2 capital P's, or whether it's heterozygous, a capital P and a little p. They'll both look exactly the same. They'll both be a purple flower. So he does a test cross on them with a white flower, which is recessive. If the flower was homozygous dominant, then 100% of the babies will be purple, because it only had, that one had dominant purple genes to pass on. But if the purple flower was heterozygous, then we'd expect half of the babies to be, half of the babies would be recessive. And that's how he could tell. And that's what a test cross is. Okay, and then we talked about the difference between co-dominance and incomplete dominance. Co-dominance, they both show up. So if you get one gene from mom and one gene from dad, they both show up. The classic example is type A blood and type B blood. If you get an A from mom, a B from dad, you have both of those proteins on the surface of your blood cells. But incomplete dominance, they blend. So if you get a red from one parent and a white from the other parent, you get a snapdragon that is pink because the two traits blended. Polygenic, what does the prefix poly mean? Many. So these are genes that are controlled, that are many, these are traits that are controlled by many genes. So the examples, skin color, can anybody else remember some? Pretty much everything is polygenic, but height is a really good example. We don't just have tall and short people. We've got all these people in between, and that's because there's many genes that are controlling how tall we are. The same thing goes for eye color, and just about everything is polygenic. There's not very many things in our world, in our lives, that are just controlled by one gene. Okay, and then sex linkage, you need to know how to do sex-linked Punnett squares. So the thing that's unique about the sex-linked Punnett squares is you have to show the sex chromosomes, and then whichever one carries the trait, you have to put the little letter as a superscript. So in this example, the Y chromosome doesn't carry the trait. So notice that the Y is blank, there's no N by it. But the X's have either a capital N or a lowercase n, and that's because the trait is carried on the X chromosome. Okay, and then gene linkage, we mentioned it a moment ago and now we're saying it again. That's the idea that some traits are actually on the same chromosome, so they are linked. Mendel didn't know anything about chromosomes, so he didn't know that actually sometimes they are inherited together because they're on the same chromosome. The part that gets tricky is that they can cross over, so sometimes they can be unlinked during crossing over. So try this puzzle. It says if the genes are on different chromosomes and inherited separately, then we would do a dihybrid problem. So we're talking about the shape of their wings and the color of their bodies. So we would expect a quarter of them would be gray normal, quarter, quarter, quarter. That's what we would expect if they're on different chromosomes. However, and so if they're on different chromosomes, we would expect a 1-to-1 to 1-to-1 ratio. What did I just do wrong? Oh, I know. Okay, sorry. But if the genes are linked on the same chromosome and inherited together, it would be more like a monohybrid problem. 50% of the parents would be, 50% of the offspring would be gray normal, like one parent, and 50% of the offspring would be black vestigial, like the other parent. But that's not at all the results we got. So it says predicted ratios. If they're on different chromosomes, we would expect a quarter of everybody. If they're on the same chromosome, we would expect 50-50. But the results that they got were actually quite divided, 965 to 944. That's pretty close to equal. But then they got 206 and 185 of these other ones. How do we explain that? Crossing over. So it says the actual results are unclear. So what's going on here? The traits are linked, but crossing over occasionally separates them. So the one that says the 1-to-1 to 0-to-0 ratio, that's closest to being correct. But then crossing over separated them from each other, so they won't always inherited together. Hopefully that makes sense. And then chromosome maps, remember determining where something is on a chromosome. We use the recombination frequency that happens during crossing over. How frequently they recombine helps us find out how far apart they are on a chromosome. So if they're really far apart on a chromosome, they're going to recombine often. They're going to get separated during crossing over often. But if they're super close together, it would be really difficult to separate them during crossing over. It can happen, but the odds are less. So then we would expect their frequency to be less. So it says here, genes that are further apart will recombine more often than genes that are closer together. Map units are purposely the distance between genes on a linkage map. One map unit is a 1% chance of crossing over. So in the example here, the color and wing genes are separated due to crossing over approximately 17% of the time. So we say those genes are placed 17 map units apart. So if the map units are closer together, then they get crossed over less often. All right, and then pedigrees, which are family trees. Males are shown as squares, females are shown as circles, and then if they're colored in this case the red ones, it indicates that they have the trait that we're looking at. The question says, is the red genetic trait due to a dominant or recessive, or are both possible? So you would maybe spend some time, maybe I won't have us spend some time right now, but it could be either one in that case. But you can come back to this if you want to and kind of do some problem solving on that. Hey, Kai, could you close the door for us just in case it gets noisy out in the hallway? Okay, what pattern of inheritance, oops, sorry, I just gave it away. What pattern of inheritance could this pedigree be showing? So remember that your brain is just going to have to think about if it's a dominant trait, if it's a recessive trait, if it's sex-linked dominant, or if it's sex-linked recessive, and play through all of some of the options here. So if I see, like if I look at parents number 3 and 4 and their son number 9, 3 and 4 don't have it, but son number 9 does have it. That sounds recessive to me. It's definitely not going to be dominant. Now, could it be sex linked? Let's think about that. The dad does not have it, so he would be X. Let's see if it's so he would be X big N Y, and the mom could be X big N X little n. Could the son get it? Yes, he could. Yeah, if it's on the X chromosome, then mom could have passed it to him, because what does dad give to his sons? If it were sex linked, he gives the Y chromosome. He gives the Y chromosome. Good. So autosomal, it says autosomal recessive. It could. Recessive. Oh, but it can't be dominant. How could it be dominant?
Well, these are the options. Those are your options. Yeah.
Oh, well, okay, there we go. Sorry, it doesn't have an answer here then. So it's definitely not autosomal dominant, and then I would guess that it's also not sex-linked dominant. So recessive and sex-linked recessive are both options. All right, non-nuclear inheritance. Remember that when we think about inheritance, we're usually thinking about the DNA that comes in our nucleus. But remember that we do get DNA that comes in our mitochondria, and that's becoming really popularized with some of these websites like 23andMe, where they'll study your DNA. They're also looking at the mitochondrial DNA because that's passed on through the female's side of the family. So you're able to look at your family history through the women based on just the mitochondrial DNA. Chloroplasts in plants. Exactly. So chloroplasts in plants also carry DNA because of the whole endosymbiotic theory. We believe that they were free living bacteria at one time. Sperm and pollen don't have organelles. They just have DNA. So remember that if an egg is like this big, a sperm is like this big, right? If an egg is, this big, a sperm is about that big. It's just tiny. All it's carrying is the DNA, but the egg has everything else in it. The egg's got the mitochondria and the Golgi and the lysosomes and all the other organelles. And so any kind of DNA that's found in the other organelles is going to come through the egg, and that comes from the mother itself.
How big is like the mitochondria compared to the egg?
Really, really, really wee also. So you can have hundreds of mitochondria in one cell. So super, super small.
If you're like, sorry, this is kind of
okay.
If you're like looking at an egg, like under a microscope, like a really strong microscope,
yep.
What could you see?
Well, it would depend how strong the microscope is. Yeah. So if it's a light microscope, it would just look like a cell. So it wouldn't look like it would just be the cell with a nucleus, and that would be all that you see. You wouldn't see the organelles. If you have a scanning electron microscope or something that's a more powerful microscope, then you can see the organelles as well. All right, non-disjunction. So this relates to meiosis. Remember, in meiosis, the chromosome number should be cut in half, and then the homologous partners should be separated from each other during anaphase. But in non-disjunction, some of the partners or one of the partners stays stuck together and they don't get separated. And so what ends up happening is you end up with egg or sperm cells that have either too many chromosomes or not enough chromosomes. And what can you name any genetic disorder that's caused by non-disjunction?
Down syndrome?
Down syndrome, right. So Down syndrome itself isn't caused by non-disjunction, but you end up with a sperm or an egg that has an extra chromosome in it. And so the kids end up with an extra chromosome 21. So the trisomies that we learned about were Down syndrome, Klinefelters. So those are boys that are XXY. So they got an extra X. Non-disjunction happened with the X chromosomes. And then Turner syndrome is X empty set or XO. That means that there was no, one of the sperm or egg did not have one of the sex chromosomes in it.
Well, what happens then?
Say that one more
What
time.
does that do to something?
Very little. So most of the time,
She's just infertile.
there you go. Most of the time, having the wrong number of chromosomes is fatal. These are the very rare conditions where having the wrong number of chromosomes is a survivable condition. And for Turner syndrome, it's very mild. They tend to be short females that are infertile, but otherwise lead perfectly normal lives. Klinefelters, remember, they develop female characteristics at puberty, but they are males in terms of their genitalia. And so that's a little bit more difficult in terms of their personal lives. And then Down syndrome, we know, everybody knows Down syndrome. Okay, polyploidy. Look at how many chromosomes are here. If that were a human, that would be terrible. That would be a fatal condition. But in some organisms, polyploidy is actually good. So some plants, frogs, and fish, it's the same advantage that female humans have, where if one of the X chromosomes isn't functioning correctly, they have a backup X. So when you have polyploidy, yeah, yes, exactly. So when you have polyploidy, then they can use the most functional chromosome, and that gives them an advantage. So it gives them sort of, yeah, whatever, we'll just say it's an advantage. Okay, we already reviewed this in our biochemistry, but it is part of heredity also. DNA structure. Nucleotides on one strand are covalently bonded to each other through phosphodiester bonds. So the phosphodiester bonds are between the sugar and the phosphate group. And then the bases on opposite strands are hydrogen bonded together. Is that a strong or a weak bond? Weak. And so we can separate the two sides of a DNA molecule very easily, and that becomes super important when we're trying to replicate the DNA. So we say that the two strands are anti-parallel. Can you see that they're upside down from each other? Can you see that they're running like this? Look at the structures. Look at the sugar in particular. Look at where the oxygen is, and then it's on the bottom on the 3' end. Okay, and then purines and pyrimidines. Purines are the ones that have 2 rings, so they're on the left side, and pyrimidines have 1 ring. The word pyrimidine has a Y in it, and so do thymine and cytosine. Not that that's that helpful. Uracil is a pyrimidine, and it doesn't have a Y in it, so whatever. But and then uracil replaces thymine in RNA. So remember that DNA has A, T, C, and G, and RNA has A, U, C, and G. Okay, so when we need to copy the DNA, it's called replication. And we have to replicate the DNA in order for the cell to divide. And this is where we get mutations. This is where the cell makes mistakes. So we say it's semi-conservative, because 1/2 of each part is saved, it's conserved, and then the other half is rebuilt. So that's semi-conservative. Helicase is the enzyme that's unzipping it. Topoisomerase is untwisting it ahead of helicase. And then the RNA primers, let's see if we can see an RNA primer. There it is, right there in red. The RNA primers are put on by something called primase. They show DNA polymerase where to begin replication. So remember, on the leading strand, the DNA polymerase is going to go very, very fast right behind helicase. It's as fast as helicase is separating the two sides. DNA polymerase is adding the A's, T's, C's, and G's to copy the DNA. But on this side, the DNA polymerase has to run the opposite direction. It does it over and over and over again, which is what creates the Okazaki fragments. And then those Okazaki fragments get joined together by the enzyme called ligase. Replication occurs during the S phase of the cell cycle. So remember, it goes interphase, prophase, metaphase, anaphase, telophase, and interphase has 3 parts to it, G1, S, and G2. S is the phase that this is happening in. It's where mutations happen. It turns monovalent chromosomes into bivalent. After proofreading enzymes, the error rate is only about 1 in 10 billion, but those add up over time. And so it's the number one reason why we have higher rates of cancer in adults than we do in young people, because there are more and more and more mutations the older that you get. Okay, transcription and translation. So that was just replication. So transcription is making RNA. So basically, RNA polymerase attaches to a promoter region in front of a gene. So there's the promoter region. It's in light green. It's often identified by a Tata box in eukaryotes. Prokaryotes don't have this Tata part, but that's the promoter. So then these transcription factors, they look like little grapes. If this is a eukaryote, they have to be there. And then the guitar pick, this white guitar pick, that's the RNA polymerase that's going to read one side of the DNA and make the RNA. It only copies one side of the DNA. So only one side is copied because RNA is single-stranded. The template strand is the side that's being copied. So that's the darker blue strand here. The red is the RNA that's being made. And the non-template or coding strand is the light blue strand that's not being copied at the moment. Okay, and then, so mRNA processing, this only happens in eukaryotes. Prokaryotes, just everything is simpler. As soon as mRNA is made, the ribosomes clamp on and they start making a protein out of it. In eukaryotes, a lot of things have to happen. It says a modified nucleotide is added to the 5' end, so we call that the 5' cap. You can see it in yellow on the left side. And then a tail of several hundred adenines called the poly A tail is on the right side. That's added to the mRNA. And then we have to cut out the introns and splice the exons together. So the introns are the sort of so-called junk. They're not really junk, but they're pieces of DNA that we're going to remove. And then we attach the exons, but alternative splicing says that you can put the exons together in different orders or different numbers of exons and put them together. So it means that one gene doesn't code for just one protein. It means that one gene can code for numerous different proteins depending on how we splice out the introns and put together the exons. We used to think one gene, one protein. We used to think that's the way it was, and now we know better. Okay, and then this is just a picture or a diagram of a ribosome. It's got 2 subunits, the big subunit on the top and then the small subunit. It's got 3 sites, the EPA. So the A site is where the amino acids come in from the left. The P site is where the polypeptide is kept. And the E site is where the trucks leave, where the tRNAs leave. Okay, and then we just need to practice what tRNA is. Those are transfer RNA molecules. So down at the bottom, so look at the image on the left. I kind of like that image the best. Down at the bottom, there are 3 letters, AAG. That's an anticodon. Those are like the letters on the writing on a truck that's driving down the highway that tells you what that truck is carrying in it. Plumbing supplies, carpeting, whatever. So it says they bring amino acid to the ribosome. The amino acids are attached up at the top. I call them trucks because they're carrying the building supplies, which are the amino acids. And then the anticodon loop is at the bottom. And it is complementary or opposite to the mRNA. Genetic code, important. It's universal across all life. So what is that an indicator of? I feel common ancestry. Oh my gosh, I just feel certain they're going to ask a question like that. I don't know if it's going to be about this or what it's going to be about, but they're going to say, oh, it's common across all these organisms. And you're going to be like, Common ancestry, that's an indicator. Okay, so mRNA is read in units of 3 bases at a time. We call them codons, and there are 64 possible combinations of those bases, but there's only 20 amino acids. So there's more than one possible codon for each amino acid. However, it's unambiguous, which means it doesn't matter what the life form is. If they have CUU, it always codes for leucine, period. No exceptions to that. It has start and start sequences. So the start sequence is always AUG in all organisms, and then the 3 stop codons. Wobble describes the redundancy in the genetic code, the idea that the same amino acid may be encoded by multiple codons. And then it says the codons that code for the same amino acids, differ only in the 3rd nucleotide. All right, on to translation. So we did transcription where we made the mRNA. Now we need the mRNA to leave the nucleus and go to the ribosome. So the start codon is AUG. The trucks pull up carrying their amino acids, pull up to the ribosome, carrying their amino acids. There's peptide bonds that form. So look at the little chain of spheres there. The little black dots in between them, those are peptide bonds. And then a polypeptide is released when the stop codon is reached. Polypeptide, remember, means baby protein or immature protein in this case. Since prokaryotes don't have a nucleus, transcription and translation are coupled. They happen simultaneously. So we call that process polyribosomes. There's the simultaneous translation of a transcript even while the transcript is still being made. So you have many, many, many ribosomes already working on the one RNA. We don't have to wait for it to leave the nucleus. We don't have to wait for introns and exons and all that stuff right away. So bacteria are incredibly efficient. They can reproduce every 20 minutes. This is part of the reason why they're so efficient. Okay, and then retroviruses. This is something College Board wants you to know about. Retroviruses are unusual because instead of having their genetic material be DNA, their genetic material is RNA. They possess this enzyme that's called reverse transcriptase that reads the RNA and then converts it to DNA. The problem is that it makes lots and lots of mutations, and there are no proofreading enzymes to fix those mutations. And so what that does is makes it really hard for us to make vaccines for it. They say that every person who has HIV has their own variety of HIV because reverse transcriptase makes so many mutations. All right, we're getting near, we're doing good here. Gene regulation refers to turning genes on or off, slowing down or speeding up transcription, or breaking down no longer needed proteins. Differential gene expression, words like that just go in one ear and out the other for me, so you have to really pause. So different gene expression, so whether the genes are turned on or off or how they're expressed, it's different. It's necessary to conserve energy. We don't want to make the enzyme for digesting lactose if lactose isn't present. And it can be triggered by either intracellular signals, so signals from inside the cell, or signals that are coming from the outside of the cell. So we learned at this time, we learned about the operons that you find in bacteria. You don't find operons in eukaryotic cells because our DNA is much more complicated, but this gives us a good starting place to figure out how DNA works. So they're organized clusters that contribute to a particular task. There's 2 types: inducible. You can induce them, which means that they're usually off, or repressible. They're usually on and you can repress them or you can stop them. And then they are both examples of negative gene regulation because repressor proteins shut them down. So the lac operon is the one that we learned about first. In this example, there's no lactose present. And I can tell there's no lactose present because this little red piece that looks like a Lego doesn't have any lactose in it. So right now it is attached to the operator, and the RNA polymerase, the guitar pick, can't get through. So it's not going to move ahead, and the whole gene is shut off by that repressor. When lactose is present, now it shows up as a green sphere. It touches the repressor and changes its shape. So now it's no longer blocking the RNA polymerase. RNA polymerase can now read across the gene, so that's DNA that it's reading, and it can make a strand of mRNA, and then the mRNA gets translated by ribosomes into, in this case, 3 enzymes. Would you need to know their names? No. They'll give you this diagram if they're going to ask you about lac operon. They'll give you this diagram to help trigger your memories. Ian?
Did the inducers just naturally disconnect themselves from the repressors after a while?
Say it one more time.
Do the inducers, in this case lactose, like just disconnect themselves after
Yes.
a while?
So as I don't know the details, but all I know is as the lactose levels start to decrease in the cell because it's all being digested, then as that level drops off, then they fall off. So for whatever mechanism that's keeping them attached, they come off once lactose levels are low. So then the repressor returns to shut the whole gene off again. It's a super good question. I don't know the specifics except when lactose levels are low, they fall off again. Okay, and then positive gene regulation. Those were negative gene regulation because we're repressing the system. Positive gene regulation is increasing the system, increasing transcription. So we learned about this turbocharger that's called CAP. So when lactose is present and glucose is low, the cell's feeling hungry, like we are right now. Low glucose means the amount of cyclic AMP is high. Cyclic AMP is that orange diamond. It binds to the catabolite activator protein, or CAP, turning it on. And that makes the RNA polymerase bind more efficiently to the DNA and makes it work faster. So then it makes the proteins faster and it speeds up the whole process. Okay, and then I'm skipping, there's a bunch of other things that are about eukaryotic gene regulation, but this one is specifically mentioned in the College Board notes. And so I just want to remind you of something called small mRNAs. There are bazillions of types of small mRNAs, but the general idea is they can do one of two things. Small RNAs can either degrade target mRNA molecules, or they can stop translation of the target mRNA. If you look at the diagram, there's an mRNA, a small mRNA has attached to the red mRNA, and now the ribosome can't read it. And so that's in essence stopping translation of the mRNA. But it's a way to shut off the gene, so that protein is now shut down. So it's a type of gene control. Okay, and then just quick reminders about mutations. Changes in DNA affect changes in protein structure and physiology. Whether a mutation is detrimental, is that good or bad? Detrimental is bad. Beneficial or neutral depends on the environmental context. So is it good to be an albino squirrel? Well, maybe if you live at the North Pole, and maybe not if you live, in the tropics. So it just depends on the circumstance. So there's two major types of mutations: point mutations. One DNA base is replaced by another, and frame shifts. Frame shifts are when we have an insertion or deletion that screws up all the frames of 3. Do you guys remember frame shift mutations? And it's screwing up kind of the groupings of the 3. Good. Sickle cell anemia, which is what this one is due just to a simple point mutation. One letter is wrong. Okay, how do prokaryotes evolve? This paragraph on the top I feel like is important. Bacteria clone themselves in the process of binary fission. Since they don't have sex that would increase variation, they have evolved alternative ways to exchange DNA with other bacteria. This is called horizontal gene transfer. Do you remember vertical gene transfers from parent to offspring? But horizontal gene transfer is from like cousins or something like that, like non-relatives even. That's horizontal gene transfer. So there's 3 ways that prokaryotes can do that. Transformation, they can just, remember the sound? They can just suck up the DNA that might be in the environment, and then they immediately incorporate that into their own DNA. Conjugation is kind of like bacteria sex, but there's no male-female. They can just exchange DNA with a non-related bacteria. Transduction is where a virus that's called a bacteriophage accidentally picks up a bacteria's DNA and then transfers it to another DNA. It seems like that's an incredibly unlikely scenario, but when you think about how many bacteria there are and how many bacteriophages there are, even if it's 1 in quadrillion chance of it happening, it's happening because there's so many of them. So I made those numbers up, I have no idea. And then transposition, a transposable element or a jumping gene is a DNA sequence that can change its position on the genome, sometimes creating or reversing mutations and altering the cell's genetic identity. So in this case, lots of times it's antibiotic resistance genes. In this case, the light blue with the gold circle, that's the that's the prokaryote circular plasmid. Circular DNA, I said that wrong. The gene can come off and go into the plasmid, so now it's the green with the yellow, and that's an antibiotic resistance gene, and then that can be transferred to other bacteria through conjugation or other ways. Okay, and then we're on our last section, which is on biotechnologies. So just reminding you, these are kind of fun. Restriction enzymes or endonucleases, that's what they're probably going to call them because they always use the ugly words if they can. Are enzymes that cut DNA at specific nucleotide sequences. So they're looking for specific letters. They can make blunt cuts or sticky ends. So it says it right there, but a number 1, it made sticky ends that can actually re-anneal with each other, or they can anneal with the target sequence. There's hundreds of different types of restriction enzymes, and they allow us to do things like DNA fingerprinting or genetic engineering experiments. So what you're seeing on the left, that diagram is a genetic engineering experiment. The gray is DNA that we're trying to insert into already existing green DNA. And as long as you have they're both cut by the same restriction enzymes, they can stick together. Okay, PCR, the job of PCR, this is a biotechnology, is to copy DNA unnaturally. So it's a machine that does it, makes bajillions, quote me on that. Please don't write that on your AP test, of copies of DNA. So for example, we have a tiny amount of DNA at a drop of blood at a crime scene. We scrape it up, we put it into a PCR machine, and an hour later we have bajillions of copies. Or we could make bajillions of copies of a gene of interest. Let's say you've been studying for 20 years a particular gene that you want to make lots of copies of, put it into the PCR machine, and in an hour you have lots and lots of copies of it. You don't need to know the steps of PCR, but this is basically what it's doing. It pulls the DNA apart. It adds primers that copy the sides, and then you add heat and it pulls them apart, and then it adds primers that copy the sides over and over and over again. It's all about temperature and annealing and whatever, but you don't need to know that. Okay, and then DNA fingerprinting, you do need to be familiar with it. So it's the idea that DNA is, you cut the DNA with restriction enzymes, and the fragments of DNA are negatively charged. So you put them in the negative end. Look at the little test tubes. They're going in the negative end of the electrophoresis machine. And then you turn on the energy, you turn on the electricity, and the positive charge, the negative charge pushes them away, and the positive charge attracts them. So the DNA fragments start moving across. Which fragments move the furthest? Which fragments get the closest to the positive end? The shorter fragments, yeah, they're going to travel a lot faster. And so over time, they're going to spread out, and the little fragments will have traveled further, and the larger fragments stay closer to the beginning part. And then this is just an example here. We have a DNA fingerprint and whodunit at the crime scene. It looks like suspect number 1 matches what the crime scene was. Okay, and then we can use gel electrophoresis for crime scene analysis, COVID research, doing genetic analysis on different types of COVID, that kind of thing, genetic disorders research, paternity, blah, 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 blah. Okay, And then the steps of a genetic engineering, we kind of went over that just a moment ago. Scientists can now remove a gene of interest from one source and add it to the DNA of another source using restriction or endonucleases. You have to use the same one on the source and the same one on the plasmid so that the sticky ends match up. And then you mix them together, and then you give the bacteria time to suck them up, and then the bacteria become transformed when they suck up the new plasmids. Use antibiotics to kill any bacteria that did not take up the plasmid, and now the bacteria are going to produce the protein that you're interested in. And you can just basically milk the bacteria for the protein that you're trying to get out of it. So examples of recombinant DNA, we've added genes for pest resistance to plants. We've added genes to bacteria to help clean up things like toxic waste spills. We've added proteins to collected to dissolve blood clots, and then human growth proteins for people who are behind on the growth charts. And then this is the very last thing, DNA sequencing is just that's actually what DNA sequencing it looks like. Each color is a different A, T, C, or G. So you, by looking at it, you can tell the A's, T's, C's, and G's. The Human Genome Project is where we first started doing it. It started in 1990, and we finished it way ahead of time because the technology improved super, super fast. So we were done by 2003. And then this is the last slide. It's just another type of sequencing method called the Sanger method. Do you need to understand this? Nope. Just be aware that lasers are used to detect nucleotide sequences. Everybody, we are all done. Does anybody have any questions before we go? Awesome. Have a great afternoon. I got to shut this one down.
That was a big sigh.
Everybody else does.
We should do
Oops, that's not what I meant to. Bye, everybody. Have a great afternoon.